Hello and welcome to Microscopic Diaries. I'm Microbiota, your host. In today's episode, we journey into the world of fecal microbial transplantation to see how swapping gut bacteria can cure disease, alter behavior, and blur the lines of identity. Imagine if a simple transplant could change who you are, not an organ transplant, but a microbial one. Picture your gut as a bustling city of trillions of tiny inhabitants. Now, introduce a whole new population into that city. The result? The city's mood, energy, and even its defensive fortifications might transform. This isn't science fiction. It's the promise and mystery of fecal microbiota transplantation, or FMT, a procedure often dubbed the Microbiome Makeover. So, what exactly is a fecal transplant? Fecal microbiota transplantation is the process of transferring a minimally manipulated community of gut microorganisms from a healthy donor stool into a patient's gastrointestinal tract. The primary clinical use of MT today is to treat recurrent Clostridius difficile infection that is unresponsive to standard antibiotics, where FMT has shown cure rates up to 90% after one or two infusions. In recent years, FMT has also been explored in research trials for other conditions like inflammatory bowel disease, metabolic syndromes, though robust evidence for widespread use outside CDI is still lacking. The foundation of a safe FMT is rigorous donor screening to exclude anyone with communicable diseases or health factors that could adversely affect recipients. Stool donors are typically healthy adults with no history of chronic gastrointestinal disorders, autoimmune diseases, metabolic syndromes, or high-risk behaviors. Screening questionnaires and interviews are used to thoroughly review the donor's medical history, medication, supplement use, diet, and risk behaviors. Candidates who pass initial questionnaires undergo extensive laboratory tests on blood and stool to detect hidden pathogens. Blood tests typically screen for blood-borne infections such as HIV-1 and 2, HTLV, viral hepatitis, syphilis, and sometimes other infections like strangulitis or toxoplasma that could be transmitted via stool. Donors are performed for stool tests for some entering pathogens, which are bacterial cultures for PCR for Staph toxin, Salmonella, Shigella, Campylobacter, Yersinia vibrio, pathogenic E. coli strains, as well as ON parasites and gastrointestinal viruses. In recent years, multi-drug resistant organisms have been added to screening patients. There happened an incident where FMT transmitted a resistant E. coli infection. Donors are also screened for emerging threats like SARS-CoV-2. Guidelines used during the COVID-19 pandemic require assessing donor exposure history for COVID-19 and testing donor samples or swabs for the virus to avoid transmission. Once a donor is approved, stool samples are collected following strict hygienic procedures. Donors are typically given a sterile collection kit to produce a sample. For home collections, donors are often instructed to refrigerate the stool immediately and deliver it to the processing lab within a short time window. In a clinical hospital setting, some donors may produce the sample on site. Timing is very important. The stool should be as fresh as possible to preserve anaerobic bacterial viability and reduce overgrowth of any contaminants. For stool that will be stored frozen, guidelines often specify processing and freezing within about 6 to 8 hours of collection to ensure microbiota preservation. If immediate fresh transplant is planned, processing usually begins within 6 hours of defecation. Personal handling the stool, wear appropriate protective gear, and work in a sanitized area. Each donation is labeled with a unique ID to maintain traceability from donor to recipient. Inside a sterile lab, the stool undergoes a transformation. Blended with a saline alkali solution, filtered to remove undigested food and fiber, homogenized into smooth microbial slurry. The final product is either used fresh within a few hours or frozen at -80 Celsius, often with glycerol to protect the microbes. frozen FMT can be stored for months, allowing still banks to prepare a normalized pre-screened doses ready to ship. Recent innovations have explored freeze-drying tools to create shelf-stable FMT powder that can be encapsulated. Lyophilized FMT involves removing water under low temperature and vacuum, yielding a dry powder of bacteria that can survive long-term at room temperature if properly formulated. So after we have a material, how we deliver it to the patient? FMT material can be delivered to the patient's gastrointestinal tract through various routes. The choice of delivery method may depend on the indication, patient's condition, and practical considerations. Key delivery methods include colonoscopy, retention enema, upper GI delivery, and oral capsules. FMT occupies a unique and evolving position in regulatory frameworks. Regulatory bodies like the U.S. Food and Drug Administration and European Medicine Agency, as well as national health authorities, have implemented guidance to ensure FMT is used safely and effectively. The field of microbiome therapeutics is rapidly evolving, and FMT methods today may be a stepping stone to more advanced therapies in the near future. Next generation microbiota therapeutics are worked on now. There's a significant interest in moving beyond transfer of whole stool toward defined microbiota consortia or stool-derived products that can be produced under controlled conditions. Researchers envision replacing crude FMT with cultured bacteria mixtures that achieve the same clinical effects but with a far greater consistency and safety. There is an old saying, trust your gut, but who knew your gut's intestinal residence might shape your mind? In cutting-edge experiments, science found that transferring gut microbes can transfer moods. In one of the studies, researchers took bacteria from people suffering clinical depression and introduced them into the guts of rats. And those rats began exhibiting depression-like behaviors. They became less motivated and more despairing in stress tests. It was as if some dark cloud of the donor's mood had traveled along with their microbes. In fact, at least 5 studies now show that germ-free or microbiome-depleted rodents inherit depressive traits after receiving transplants from depressed humans. And it's not just depression. Even complex disorders like schizophrenia appear to have microbial fingers in the pie. In 2024, a team transplanted stool from patients with schizophrenia into mice. Astonishingly, the mice developed schizophrenia-like symptoms. They became antisocial, showed an abnormal hyperactivity, and even their brain gene activity changed. And also, similarly, fecal transplants from children with autism spectrum disorder have been explored. In one recent clinical study, about 98 children on the autism spectrum received FMT and showed notable improvements, not only in GI symptoms, but also in co-autism behaviors and sleep over the following weeks. These are preliminary results and need a validation, but they paint a poetic picture. By changing the invisible ecology inside us, we might ease a troubled mind. As we wrap up, the story of fecal transplants leaves us marveling at a profound concept. Our identity is partly a colony. We humans are not lone organisms. We are walking ecosystem. Changing that ecosystem, giving ourselves a microbiome makeover, can heal and even subtly change us. In this episode, we have seen how FMT cured disease and surprisingly shifted traits, lifting moods, taming guts, trimming or expanding waistlines, tweaking immunity. It's a reminder that mind, body, and microbiome are inseparably intertwined. There is something profoundly poetic about the notion of a fecal transplant, poetic in a practical, earthy way. It transformed the old saying, one man's trash is another man's treasure, into a medical reality. One person's waste becomes another person's remedy. In the grander shame, it blurs the line between self and other. A piece of you can heal me. My microbes can become your medicine. In a world that often emphasizes individualism, the microbiome reminds us of our deep interconnection. Thank you for joining this journey through the hidden world inside us. In the end, the humble fecal transplant teaches us that solutions to big problems can come from unexpected places, and that sometimes to renew life. You have to get your hands just a little dirty. Stay curious, stay open-minded, and remember, even in the strangest corners of science, there is wonder to be found. Subscribe to our YouTube channel, follow us on Instagram, like this video if it sparkled your curiosity, and leave comment. Until next time, stay curious, stay kind, and always listen to your gut.